Okay, good morning. Time to shift gears a little bit to a disease called ITP. I'm going to be talking about genes that are influencing the development and severity of chronic ITP that we identified through whole exome sequencing. I'm presenting this on behalf of the ITP Consortium of North America. So just a brief background about ITP. This is an autoimmune platelet disorder that affects children and adults, about 1 to 5 per 100,000 prevalence. It can result in a range of bleeding symptoms from mild to life-threatening. Treatments are very expensive and they have variable success rates and safety profiles. A lot of them have high toxicities. Basically, what happens in this disorder is you have platelet destruction, not due to a problem inherent with the platelet, but due to the fact that the immune system has become dysregulated and recognizes the platelets as foreign. And many immune abnormalities have been described to contribute to this problem, including IgG-mediated platelet destruction, abnormal platelet production in the bone marrow. But for this talk, I'm going to focus on the new emerging evidence for the T cells being players in this disorder. So why genomics? This has not been historically thought to be a genetic or heritable disease, but there's clear biologic heterogeneity, very different presentations, very different clinical courses for this disorder. So a lot of people have sought to investigate genetic changes that might affect different aspects of disease presentation or course. But the studies that have been done so far have been limited by small sample sizes, candidate gene approaches, and lack of validation. So our hope is that we can, with a bigger study, improve understanding of how ITP happens, improve understanding of stratifying patients by risk, whether they're likely to spontaneously remit or become chronic, and also identify what treatments may be best for a given patient and thereby avoid toxic treatments that would be unlikely to be beneficial. So the goal of our study was to utilize next-generation whole exome sequencing to identify variants that may affect disease predisposition or severity. Our pediatric chronic ITP cohort was a total of 262 patients. 173 came from the North American Chronic ITP Registry. 89 came from the Platelet Disorders Center at Cornell. 83% had primary ITP, which means that it was not triggered by another underlying autoimmune disorder. The mean age of the cohort was 8 years. For our study, we focused on the Caucasian non-Hispanic subgroup, which was 172 patients. And the reason that we chose to do that was because we have very robust control data, including the atherosclerosis risk in communities cohort out of the University of Texas, which is all European American healthy volunteers, as well as publicly available database controls. So by selecting only the Caucasian patients, we eliminate the wide variability in genetic diversity that comes from different ethnic backgrounds. So the first thing we did was compare our Caucasian ITP cases to the 5,664 matched controls in the atherosclerosis risk in communities cohort. And what we found was 188 total damaging mutations, meaning they resulted in a significant problem in coding. And the most significant finding is up here at the top, if you can they see the arrow? Okay, so in the IFNA 17 gene, which is responsible for TGF-beta signaling and T regulatory balance. T regulatory cells normally, their job is to prevent inappropriate immune activation. And so by abnormal Treg function, you have increased immune activation. And so we found in this gene, in our 172 cases, 91 minor alleles or 91 abnormal codes out of over 4 variants, compared to 17 minor alleles in 5,664 controls, for a p-value of 4 × 10⁻13. The other genes on this list are all involved in some way in either increasing immune activation, or decreasing self-tolerance, all favoring heightened immune activation, which is known to be an important factor in ITP. So again, just to highlight the most significant finding of the study so far, this IFNA 17 gene, interferon alpha 17, again, it's got a known role in T-cell activation. 43% of the ITP cohort had at least one IFNA 17 variant. The most common variant was found in 26% of the cohort, one single variant, and that was an isoleucine to arginine change at position 184. This was present in only 5.6% of controls. We also identified 2 patients who had a variant in IL-17F, interleukin 17F. This is one that has been previously described in the literature, although this is a different amino acid change. And this was found in 2 of our patients and none of the 5,000 controls. The next thing we did was a gene set enrichment to try to determine if this these mutations that we found could be linked to a certain pathway, and maybe multiple small variants in several genes could actually be linked to one pathway causing a much bigger problem. So we found 5 genes with an enrichment score of 0.96, and those are listed here. And this was involved in a pathway called toll-like receptor signaling pathway. The next thing we did was we wanted to try to stratify the patients by disease severity. Again, we talked about the wide heterogeneity of the disease presentation. And so we isolated the 106 patients in our cohort of 172 who required a second-line therapy, meaning that their first-line approach to treatment failed. Therefore, they needed a more aggressive approach to their treatment. We isolated them as a more severe disease phenotype, and then we ran the same analysis for just those 106 patients against the 5,000 controls. And we were looking here for variants that were present only in that cohort and not in those who only required frontline therapy. And you'll see the 4 genes listed here were the ones that were significant in that cohort and not in those who did not require second-line therapy. The most significant of these findings is DOC3, that's the one listed at the bottom, that's involved in toll-like receptor signaling again. We found 5 variants over that gene. And in this minor allele count area, you'll see that out of the 106 cases, we found 28 minor alleles. We only found a total of 31, so there were only 3 in the cohort who did not require second-line therapy. So 90% of the findings were in the people who required second-line therapy, so that's a significant difference. And again, this is compared to only 26 out of a total of 5,000 controls. So what's the take-home message here? Why are these findings important? So this is the first time that we have used whole exome sequencing to look for novel variants that might predict disease predisposition or chronicity in patients with ITP. We identified important coding variants, damaging variants in T-cell pathways. And the hope is that these findings could ultimately help us understand ITP biology better, help us stratify risk for patients, whether they're likely to remit or become chronic, and also to help them help us stratify treatment according to which treatments are likely to benefit the patient, and again, avoid toxic therapies that are less likely to be beneficial.